Welcome again. This is the Sight and Sound Bites webinar series. Thank you for coming. This bi-weekly webinar series presents the research at the University of Pittsburgh School of Medicine. Today's topic is seeing the future of gene therapy, or we should say, I guess, seeing is the future of gene therapy. But I'm Lawton Snyder, CEO of the Bioengineering Foundation. The Bioengineering Foundation supports research to advance care for vision, hearing, balance, voice, and cancers of the head and neck at two of the world-renowned departments of ophthalmology and otolaryngology at the University of Pittsburgh. The funds we provide from the Bioengineering Foundation to support research are really only made possible because of philanthropic support. And we have certainly benefited from a lot of very generous support over the years, and I can tell you that what we're seeing is the result of that, where we have one of the stronger research programs in ophthalmology, really in the world. So our housekeeping, our chat is disabled on this Zoom presentation, but we will use the Q&A function. So if you have a question at any time, click on the Q&A function, and you'll be able to answer your question, ask your question, and we will answer those at the end. Please refrain, if you can, from asking personal health questions. You can certainly send questions to Craig Smith, whose email you have with your invitation. What I may do is hold any personal health questions also to be answered by email if you put them into the Q&A function. Please feel free to use the subtitle function with closed captioning. That's what it's there for. And we will be adding you to our mailing list for future webinars. You can always opt out if you like, but we have lots of good programs. I think you'll enjoy them. We have three outstanding presenters today, and we also have our distinguished chairman, Dr. José-Alain Sahel, who's a distinguished professor and chairman of ophthalmology and the Bioengineering Foundation Endowed Chair at the University of Pittsburgh School of Medicine, and the director of the UPMC Vision Institute, an exceptional class professor at Sorbonne University in Paris. He'll be joined by Dr. Leah Byrne, PhD, who's an assistant professor of ophthalmology at the University of Pittsburgh School of Medicine and runs the Gene Therapy for Retinal Disease Laboratory. And then Michelle Alabek, who is a certified genetic counselor and coordinator of the Retinal Dystrophy Clinic. So I'm going to go ahead and ask our group to get started. They're ready to go. I think Dr. Sahel was going to first start off by saying a few words and then introducing the program. Thank you, and Dr. Sahel.
Thank you very much, Lonnie. Welcome, everyone. It's an update because I think we had a webinar on that topic a few years ago, but many things have happened in the meantime. And the goal of today's webinar is to focus on what is going on currently for the patient we are seeing in our clinics, and also the prospect of new therapies that are upcoming already in clinical trials or will be in clinical trials very soon. So we'll have today with us Michelle Alabek, who is managing the Retinal Dystrophy Clinic, who is a genetic counselor, but is also, for many of you, the main contact person, the one who is putting together everything. So she's going to talk to you about how we organize the clinic and what is currently happening for patients with genetic disease in Pittsburgh. I'll present briefly a quick overview of where we are in gene therapy. And we are very fortunate. We have Leah Byrne, who is in the department and is leading a lot of breakthroughs in gene therapies, new innovation, new therapies, new vectors. And she will talk to you about one project that is especially promising. We could spend hours to speak about gene therapy, but we'll try to focus on what is indicative of the future we are living currently. So without further ado, I will hand over to Michelle for the next part of this webinar.
Great, thank you, Dr. Sahel. So I'm going to kind of start us off with really, talking a little bit about what the patient journey would look like for a patient who might be eligible for gene therapy, kind of what goes on the back end for us as far as identification of patients as well. But just wanted to start, the concept of gene therapy, why is it such a challenge? And if there's, a specific disease that we want to target, and we're just trying to get that treatment to the patient, But, there's a lot of complexities, and I think you'll hear from Dr. Sahel and Dr. Byrne at their section of this presentation, the complexities about getting the right gene to the right place at the right level and without any unwanted outcomes or challenges. And there are a lot of things that can affect that. And so I won't go into all of these details, but again, just to kind of highlight that it is a very complex and challenging topic to develop these therapies. But what I'm going to focus on is really, once we have a therapy, how do we go and get that therapy to a patient? So the first thing that we need to do is really identify who our eligible patients are that might be interested in receiving a new therapy. So one thing that's really important to know is that any of these therapies, these gene therapies or these novel therapies that are coming up for genetic conditions, each therapy has its own kind of criteria that a patient must meet. And so it's really important that we're aware of what those criteria are. There are things that we call inclusion criteria, which are things that must be met by a patient in order to be eligible for a treatment. These can include things like, what is the individual's age? What is the specific clinical diagnosis that they have? Oftentimes, the visual acuity of the current state of disease can be very important in determining if someone is eligible. Genetic testing and the result of that genetic testing is often a very important factor. And then there, again, depending on the treatment, might be very specific results on different ocular tests that are done. So for those of you who may be patients in our clinic, you know that we do a lot of testing every time that you come in. And the value in doing that is that we're able to have that data on file for you, which helps us to assess potential eligibility as new therapies may become available. In addition to what we call inclusion criteria, there's also exclusion criteria. And these are things that, even if you meet all of the inclusion criteria for a treatment, all of the things that you have to meet, if you meet any of these things, then the treatment might not be a good option for you. So things like systemic disease, if you have a cardiac history, in some cases that might be an exclusion from treatment, or any kind of contraindication to a surgical procedure. Certain medications that you're taking could make you a poor candidate for a treatment. Other eye conditions, so things like glaucoma or really high myopia, those are factors that could also make someone potentially ineligible. So again, as clinicians, what we're always doing is really understanding for each given treatment, what is the criteria, and then trying to identify patients that might be eligible. So what does that process look like? Again, it starts with our clinical team really reviewing the eligibility criteria. What things do we need to know about a patient, and what things do we need to make sure a patient doesn't have before we can approach them about a potential new gene therapy opportunity? We maintain a database of all of our patients that are seen in clinic so that we can easily run that report based on what the criteria is for that given treatment. And then as a team, we look at it and we assess, based on the data that we have available for the patients that are known to us, who might be eligible? Is there data that we don't have on a patient that might help us determine if they're eligible or not? And then ultimately, for patients that look like they would be good candidates based on the data that we do have, we will contact them and offer to arrange an updated consult visit or an updated clinical appointment to come in and talk to the doctor about what this opportunity may be. We may at that point need to also perform some updated evaluations. If it's been a while since you had a certain test, we might need to repeat that before we know for sure that you would be eligible. There might be a specific test that we need to determine if you're eligible that we haven't yet performed on you, and so we may need to do some new testing as well. And then ultimately, we kind of review all of that data again as a clinical team for anyone involved in that particular therapy, and determine if each patient is an eligible patient or not. Of course, throughout the process, we're continuously updating the patients and keeping them informed about where things stand. And it ultimately is the patient decision if that's something that they want to move forward with. Once we have identified a patient who might be eligible for a treatment, then comes the next step, which is really coordinating that treatment for the patient. There are some complexities that go into coordinating a gene therapy treatment. This picture on the right kind of maps out, there are many stakeholders that are involved in this process. There are the patients. Obviously, there are the clinicians, not just the physicians, but if it's in the OR, individuals like anesthesiology, various testing individuals that would need to be involved, genetics, low vision. And then there's kind of the operational component, always the financial piece. And with gene therapy, cost is always a topic that comes up. And lastly, the manufacturer who's making the drug. And it really takes kind of all 5 of these groups of individuals working together and being on the same page to successfully be able to treat a patient. There are also, a lot of hospital-wide implications. There really needs to be a sound infrastructure in place to be able to get these therapies to patients. And we're fortunate that at the New Vision Institute, there was a lot of forethought put into ensuring that we can deliver these types of therapies in a very efficient way, with, involvement and kind of coordination between pharmacy and OR and the clinical team in real time. And where all of those aspects are located allows us to do that in a very efficient way that many other centers may not have the capability to. We also have to start thinking, as more of these therapies are becoming available and we're offering more of these therapies to patients, how does that impact kind of our volume, our capacity for clinic appointments, our space in the OR? These are all things that we're thinking about as more and more therapies are going to become available. And again, financial considerations as well. And another important piece really is access. We can have these therapies and we can be able to offer them, but can patients access them? And there's a couple areas that are important to think about. First, just are patients aware? So we try to make patients as they come into clinic that, aware of what's coming down the pipeline. But patients that maybe don't follow with a specialty center may not have awareness that these types of therapies are potentially an option for them. The cost of treatment, as I mentioned, these drugs can be very expensive and often are covered by insurance, but when we talk about kind of equity and fairness, in the United States and globally, I think access for these gene therapies is a very important topic that we need to make sure we're paying attention to. And lastly is just kind of where these therapies are delivered. So there may be only a handful of centers, in the United States that are participating in a trial or are approved to deliver an FDA-approved gene therapy. And so you have to start thinking about, geographic access as well, and how patients might, have location be a barrier, and what we can do to kind of help overcome and get eligible patients to a center for treatment if that's what they're interested in. So I want to move then, once we would identify that a patient is eligible for a treatment, and this is focusing more on, outside of research trials, outside of clinical trials, more of an approved, FDA-approved gene therapy. One of the first things that we spend a lot of time doing is really getting all the financial approvals in place, doing a benefits investigation, making sure we understand if it's in network or out of network, getting authorizations and contracts in place, so that we're removing that financial barrier to treatment. I will say that often is probably the most lengthy part of the process. Once we have all the financial approvals in place, we would move on to kind of the pre-treatment stage, where we will bring a patient in for baseline clinical evaluations. This is really important and often can be quite lengthy, but is very important because it establishes a baseline for us prior to treatment, and then we're able to repeat that after the treatment to try and understand the potential benefit that the patient may be experiencing. Obviously, before moving forward with treatment, there would be an informed consent, and then some behind the scenes for the hospital about how we're going to go ahead and actually get the drug on site. Once all of that is in place and patients decided to move forward with treatment, there are a lot of variables in how a treatment might be delivered. It may be in an outpatient setting. Some gene therapies, not necessarily for ophthalmology, but in other settings, may be done in an inpatient setting. It could be done in an OR, or it could be done outpatient in a clinic. Again, these will all depend on the actual treatment or the therapy that's being discussed. The type of anesthesia, how long the procedure is going to take, and then what medications might a patient need to be on either before treatment or after. And then post-treatment, there are a lot of clinical follow-ups. As I said, we want to repeat what we had done prior to treatment so that we have a comparison of how the therapy may be impacting the patient. And then for patients that potentially are going to regain vision as a part of this treatment process, vision rehabilitation is something that's very important post-treatment as well. So I just put together kind of a quick schedule example. And so if we had a patient that was going to be treated today as their treatment day, you can see that we would start, and this schedule is really only starting with after we've gotten all the financial approvals in place. You can see about a month prior, there are things that need to occur, and then various visits throughout the next few months, really through the end of September at least, with then, of course, longer term follow-up, but probably on a further schedule. So not as frequent that someone would need to come back, but it is a lot of visits, and it is a lot of a lot to ask of the patient to come to the clinic as many times as we do. But again, it is all clinically really necessary. And then I just want to take a few minutes to talk about kind of what the role of a genetic counselor is. So in general, genetic counselors, primarily in a traditional setting, are really helping to facilitate genetic testing. But as a profession, we're trained to really help people to understand and adapt to medical information and related to their genetic disease, as well as the psychological impacts that may have. And I think we're learning as a profession that really is kind of translating into this gene therapy space and helping patients to navigate that process. So anytime that I'm interacting with patients that are considering a gene therapy, I put together just kind of a list of some things that I think are important for patients to consider if they might be eligible. What are some of the anticipated outcomes? Is this a treatment that we expect to stop progression of disease, or is this a treatment that we expect to restore vision? Those are two very different outcomes, and important to know going into a treatment, I think. Obviously, what the risks are. An important thing is that just because a therapy may be available, it doesn't mean necessarily that you will be eligible for it. And again, that comes back to what the inclusion and the exclusion criteria are for an individual treatment. I think it's important to consider the schedule. So as I gave that example, it is a very cumbersome and can require a lot of time commitment from a patient. And it's important to think before you agree to do something like this, that you really understand what that is. Cost and coverage, which again, we do our best to really get as much of that information addressed up front. And then, there is still uncertainty of some of the long-term outcomes and the long-term risks that might be associated with these treatments. Important to know what your other available options may be. Are there other therapies that are coming down the pipeline for this condition? How do they compare to each other? Are they similar? Are they different? And talking with your clinician to really understand and make the best choice for you. And then really focusing on, what is your available support system? As I said, the decision to move forward with one of these therapies and then the actual process of coming to all of these clinic visits, it can take an emotional toll, and kind of assessing before you go into something like this, what your support system looks like, I think can be very helpful as well. So that's all that I have prepared for today. I think now we're going to turn it over to Dr. Sahel, and then I believe we're saving questions for the group at the end.
Thank you very much. So I'm just going to give a quick overview of where we are currently in gene therapy, but obviously I won't get into too many details. We started with Michelle's presentation because we are now in a stage where we are starting to treat patients with gene therapy, which is something we were hoping to see happening and is now happening, but unfortunately not to everyone. And I want to focus on why we are not yet in a situation where every patient with a genetic disease can access to gene therapy. As a reminder, the goal of gene therapy is to correct a gene defect. So it's using what is called a vector, and you will have a word expert of vectors talking to you with Leah in a short moment. A vector is a modified virus that is containing the code for a protein that is going to be used as a therapy. So a vector gets packaged and that gets inside the cell. And from there, once it gets inside the cells, the protein gets expressed, and this is used to correct the gene defect. And there are many ways to do that, but we'll focus on a few of these. The vectors are delivered in different ways to the eye. The current most classical way to do that is to inject under the retina. So it's a schematic. So this is not exactly how we do that, but it's a surgery where we get inside the eye, and a surgery that has been used for many other conditions over many decades already, whereby we are injecting under the retina of the vector to target the cells in the retina. So on the schematic, the retina is figured in gray. It continues with the optic nerve, and the vector is injected under the retina. There's another approach, which is injecting directly into the vitreous, which is the cavity of the eye, the main actually part of the eye, which is a gel. And this is used for some of the therapeutic approaches and is mostly efficient to target the inner part of a retina. But now, thanks to the work of Leah and others, it's possible to target many other cells. And there's a new approach that has been developing over the past few years. It's called suprachoroidal. You see that in green here, where the vector is injected actually under not only the retina, but the choroid, which is the nurturing tissue under the retina. So there are three approaches. Each of these has advantages and disadvantages. Some of them are a bit more invasive, like subretinal injection. Some of them lead to more inflammatory responses, like intravitreal delivery, but they have also advantages in terms of safety and efficacy. Currently, if you look at gene therapies, there are two main indications. One is age-related macular degeneration. And the main goal of people using gene therapy is not to correct the cause of the disease, because we don't actually know precisely the cause of the disease, but to treat the complications of the disease, namely the atrophy of the retina or the proliferation of new vessels under the retina. And the goal is to limit the number of injections. Currently, patients that are benefiting from the existing therapies have to come on a monthly basis almost to get injections. Even the new therapies that have been approved in the US last year for the atrophic form of age-related macular degeneration require repeated injections. So many people are trying to find a way to limit the need for repeated injections, and there are several clinical trials underway to do that seem promising, but at the same time you want to be able also to control when you don't need to treat anymore, and this is something we don't have really at hand currently. So these are all the existing trials, and we have several in Pittsburgh. For example, Ascent, which is delivering anti-VEGF, anti-angiogenic approaches. And this is a trial that José-Alain is conducting in our department. And it's developed by a company called REGENXBIO. I don't have any interest in this company, but they are a very innovative company. And they are developing two approaches. One is subretinal, one is suprachoroidal. And the goal is to deliver a protein that is going to block the vessels that are growing under the retina in age-related macular degeneration. So this is an ongoing trial led by José-Alain in the department. But the main focus we have for many of us, Michelle, Leah, myself, is really genetic disease of the retina. And we are trying to target the various stages of the disease. So I'm going to go through this slide, spend some time on that. In retinitis pigmentosa, the cells that are targeted by the main mutations are in the depth of the retina. They are called photoreceptors. They are capturing the light and they are transforming the light into an electrical signal. These cells are affected by mutation, mostly the cells that enable the dark adapted vision, the way we have to see during the night. And this is leading to loss of dark adapted vision. But over the course of time, there is also a degeneration of what we call the cone photoreceptors that are responsible for peripheral vision in lighted conditions and also for central vision and color vision. And the cones, when they degenerate, this leads to the loss of peripheral vision. Eventually, the central field becomes more narrow over time. We also lose color vision. And over the course of time, this can lead to total loss of vision in some of our patients. So the therapies are trying to tackle the stages of the disease. There is no one single therapy doing everything. The ideal therapy is to correct the gene defect. If you are able to do that early enough, then you stop the course of the disease and the patient can potentially recover or keep a very useful vision. So this is gene replacement, which is really adding the gene that is missing, or gene editing, which is trying to correct the typo in the genetic code that is leading to the disease. Another approach is to protect the remaining cells, and this is the one I've been working on for many, many years, and actually Leah has also been involved in that for many years, is trying to protect the remaining part of the vision using trophic factors. We'll speak briefly about that in a moment. Another one is to reactivate the remaining cells of the retina to make them responsive to light and to be able to really trigger light responses in a blind retina. This is optogenetics, but it is also something that is done with other approaches like cell therapy and prosthetics that don't use gene therapy. So I'm going to really navigate briefly through that in the coming minutes. This is the team, and you heard already from Michelle. These are the people working with me in the department. On the clinical side, Boris Rosin, who is an expert in also retinal degenerations, and the two surgeons, Yossi Martel and Jay Shablani, that are helping, but also Kunal Dansingani, who is involved in some of his gene therapy projects. We see the patient and we try to identify the gene defect and then try to correct the gene defect. Currently, only a small number of patients can be really treated with these approaches. And so this is, we talk about the main one, which is currently Luxturna for the Leber's congenital amaurosis, very early onset disease that is occurring in children, leading to early onset and loss of vision. So this is Luxturna. Luxturna was approved in 2017 in the US and 2018 in Europe. It was developed by the team of Jean Bennett in Philadelphia and has been able to demonstrate a very significant effect on vision, dark adapted vision, especially navigation in dark adapted vision. And this has been one of the first gene therapies approved across the world, and the US being the first country for that. We now have approval in Pittsburgh, and we have started to treat patients with that, and we are very happy about that. But we have to remember that it's only a small number of patients. So it was developed by a company called Spark Therapeutics, licensed to Novartis in Europe. And Spark was acquired by Roche, a big pharma also. So this is available for our patients. And we spend a lot of time. We have someone who is helping us a lot in the department, Dana, and actually also the company to really make sure that every patient has access to the therapy. But we have to remember it's a very small number of patients, which means that there are many patients that currently don't have access to gene therapy just because these therapies are not yet validated. And the reason is that there are many gene defects. If you look at retinitis pigmentosa alone, we are talking about 70 different genes, and each of these genes has different gene defects. So there is a complexity and a large number of mutations. We still don't know about all the genes. We still have a few genes that are not yet identified, probably between 15 and 30% of these. And also we need to know about the stage of intervention because there is a stage where it's too late to intervene and to have a benefit of this patient. But as you can read from that, being able to have a genetic diagnosis is extremely important. This is why we emphasize so much genetic diagnosis. And we got massive support from Foundation Fighting Blindness through my retina tracker to provide genetic diagnosis for many patients, although some health plans also provide this type of coverage. This should lead to new therapies, but currently most of these therapies are in development in the laboratory or in clinical trials. This is what you can find on the website of clinicaltrials.gov, which contains a lot of information about existing gene therapy trials. As you see, some of these are for age-related macular degeneration, but there are also some of them active for retinitis pigmentosa, especially the X-linked retinitis pigmentosa, with several trials ongoing on that. Among the approaches we have been developing, because we are concerned about the fact that we may have to treat our patient a bit later in the course of the disease and not always early enough to be able to correct the gene defect, is to trying to protect the remaining vision. What patients have lost, the peripheral vision, may not be rescuable, but they have still left to avoid the total blindness. And my team has been working for many, many years on trying to protect the central vision, the cones, and the light adapted vision. And we were able to identify a key mechanism for that, where actually there is a crosstalk between the rods and the cones, the photoreceptors, and they are able to maintain photoreceptors viable. And this therapy is now in clinical trial. So I have an interest in this therapy, I have to say. And this therapy is using the endogenous mechanism for the survival of cones. So I won't get into too many details, but we are trying to rescue actually the cones by providing the signaling that is underlying their survival. And this is now in clinical trials. This is the result we had in animal models where we could rescue photoreceptors and show that you can keep the part of the photoreceptors that is responsive to light. And now this is in clinical trial, and actually Yossi Martel, who is on the slide, is the principal investigator on that. I'll move the screen here so that you can see him. He's the principal investigator, and this is in clinical trial currently, both in Paris and Pittsburgh, and actually a few centers are opening in the US, like Miami, in the coming days. Another approach is to reactivate the remaining cells in the retina. This is what is called optogenetics, where we express in the remaining cells of retina proteins that are sensitive to light, either the remaining photoreceptors or the innermost cells of the retina, what we call the ganglion cells, for example. And this is what we have done in a gene therapy trial, but we did both in Paris and Pittsburgh and London, where we were able to actually reactivate the ganglion cells, the remaining cells in the retina, and using a medical device to stimulate that, to obtain in our patient through a study that was at different doses. And again, Dr. Martel was the principal investigator of the study. And we were able to demonstrate in the first patient and now in other patients that we were able to move from being totally blind to being able to see objects, to count objects, and to locate objects. And we were able to show that this is correlated with reactivation of the brain in the visual area. So I show you an example of one patient treated with this approach, not the patient that was published, where she was totally blind and she's able to count how many pens are on the table. So she sees, as you see, 1, 2, and 3. As you will see, she will 1, 2, and then 3. And also the edge of the table. And then she's going to be asked actually to grab an object on the table. And she does that quite precisely. So now we are putting together the data from the study in many other patients. And we are actually working together with Leah Byrne, Morgan Fedorchak, and Valentina Emiliani in Paris on improving this therapy to provide much better vision than what we have currently. And this is very promising. We are also working with other companies that are developing this approach to get in the clinic. So to go back to what I showed you, gene therapy is moving into the clinic and very promising. We have to keep checking the safety of this approach, work on the various stages of a condition. We always have to ask ourselves, are we trying to just stabilize the disease and keep whatever is left for the patient, or to cure the disease? Are we trying to treat the cause of a disease or a common pathway in the course of a disease? Are we protecting the cells or restoring vision? How do we demonstrate the activity and the efficiency? And how do we demonstrate that this is what we call functional vision, which is the activity of daily living? And as you know, we had presentation on the street lab and the driving simulator in the previous webinar, so I won't get into that for the time being. So very promising era, but a lot to come. And one of the most promising projects is going to be presented to you by Leah Byrne. So I think she's going to share our screen. Thank you very much.
So first of all, thanks so much for having me. Thanks to Lonnie for inviting me to give this talk today. Yeah, really happy to have this opportunity to present just in some detail about one of the projects that we're working on, as Jose mentioned. So this is going to be a little bit more of the nitty-gritty of how one gene therapy is being developed preclinically, and hopefully we're moving this toward the clinic. I'm going to try to keep my eye on the chat box here. So please, if anybody has any questions, please feel free to pop that question in the box. And I always find it a little bit disorienting not to be able to see anybody. So please do reach out with questions and I'll know you all are out there. So yeah, I'm Leah, just introducing myself. I'm an assistant professor in the Department of Ophthalmology, and I've been working on gene therapy for retinal degeneration since my PhD era. And I'm going to tell you guys today about work we're doing to develop a gene therapy for one specific type of inherited blindness, which is called PRPF31 retinitis pigmentosa. And I just want to introduce you to the student who did a lot of the work on this project before we even begin. So medical student named Xiaohan Qi did a lot of the work that I'm going to show you here today. So as Jose mentioned, but never hurts to repeat, the way gene therapy works most of the time is that we take some kind of a vector. Most often in the retina, it's a viral vector. We empty out the genetic material from the virus and replace that with a therapeutic gene. And then that virus gets injected into the eye, where the virus can then infect cells in the retina and carry in that therapeutic gene. That gene is actually the blueprint for a protein that is used then by the retina and can hopefully restore vision in that person. And so there's a lot of different types of gene therapies out there, and Jose already described all of these, but sort of typical gene therapy that we talk about a lot is gene replacement, where you have a mutation in a gene and that's causing disease, and we just go back in and provide a healthy copy of that gene. So we just replace the mutated gene with a healthy copy. And then there's optogenetics, which Jose talked about, where we can create artificial photoreceptors. And then sometimes we use genome editing to rewrite those mutations. So this is a new tool that's been developed where we can actually, instead of replacing a gene, we go in and just rewrite the mutation in situ. So today I'm going to be telling you about a gene replacement strategy for PRPF31 RP. And I just wanted to mention really quickly that the virus that we're talking about in these experiments is called adeno-associated virus. Just want to take a minute to talk about this because we've all had bad experiences with viruses over the past few years with COVID, for example. And I think it's worth mentioning that, the virus we're working with here is actually, it's non-pathogenic, so it doesn't cause disease. So it's something that we actually live in harmony with most of the time out in the world. And the virus is very safe because it's replication incompetent, meaning it can't make more copies of itself. That's because we took out all the genetic material from the virus and replaced it with a healthy copy of a gene that we wanted to treat the patient with. But this virus has the ability to infect cells in the retina really efficiently. And so it's a really good option for retinal gene therapy. And the other thing is that, so when you deliver a gene with a virus like AAV, it leads to permanent expression. So the promise of gene therapy really is that you'll get a permanent solution to the problem after a single injection. So in theory, if everything works as we hope it will, then you will deliver the virus one time, and then that therapeutic gene is going to stick around in the retinal cells for the rest of the life of the patient. So I think gene therapy holds a lot of promise for that reason. So PRPF31, what is it? It's a very interesting disease. It's caused by mutations in PRPF31, this gene, which is basically the blueprints. It encodes for a splicing factor. And so the splicing factor is something that is required to process mRNA. So if you guys have biology backgrounds, you'll have heard of the central dogma where you use mRNA to make protein. And so it's depicted in this picture right here in this red bubble. So there's a lot of different types of mutations in the specific gene that can cause a disease. So there's a lot of different types of mutations on the same gene. And it is a rare disease, but it's one of the most common rare retinal diseases that accounts for about 2.5 to 10% of autosomal dominant types of retinitis pigmentosa. And currently, we don't really have a lot of treatments for this disease, so it really represents a major unmet need in the medical field. And right now, we're really limited to just monitoring disease progression. But as I'm going to show you, I think gene therapy holds a lot of promise for treating this disease. So one thing you kind of need to understand about the disease is sort of interesting. It's caused by something called haploinsufficiency, which I'm going to try to explain really quickly here. So everyone carries 2 copies of the PRPF31 gene. And if you have a mutation in just 1 copy of that gene, then you can either have the disease or be normal. And whether or not you have the disease is determined by if that second copy is a high expressing copy or a low expressing copy. So if you inherit from your parents 1 mutated copy and you have a low expressing copy on the other allele, then you're going to have disease. So that's called haploinsufficiency. But that also means that it's sort of a naturally occurring model of how gene therapy could work. Because if you inherit 1 mutated gene and the second copy is high expressing, you do not have disease. So if we're able to just increase the amount of PRPF31 that we're making by a little bit, it means that we're very likely to be able to treat this disorder. So again, it's just a naturally occurring model of how gene augmentation is likely to work for this disease. So it's really a promising disorder. Now, one of the biggest problems in the field is that we have not had an animal model in which to test these gene therapies. And that's because most of the time we're doing research in mice. You guys have all heard about experiments that are done in rodents in order to lead to clinical trials for people. But we have not had a mouse model. And that's because if you have 0 copies of PRPF31, it is lethal. Mice do not survive. But in mice, that second copy of the gene tends to be high expressing. And so if you only give them 1 mutated copy of the gene, they just don't have the disease, or they have very mild forms of the disease. And so we haven't really had a mouse model in which to test the gene therapies. And that's a really important step for preclinical investigation of any new gene therapy. So what my group has set about to do was to create a mouse model of PRPF31. And we actually used this thing that I mentioned before, CRISPR, which is basically a pair of molecular scissors that we can use to cut the genome. And so we put CRISPR into these mice and we cut out a little piece of the PRPF31 gene. And by cutting out that piece of the PRPF31 gene, we inactivate it. And then that leads to disease in the mice. And we delivered this, then we delivered a gene therapy using a virus on top of creating this animal model. So I'm going to walk you guys through some of the actual data that we've produced in my lab, and showing you how we've been able to create this mouse model, and then showing you some of how the gene therapy has efficacy in the mouse model. So we actually used a virus to deliver these molecular scissors, this CRISPR, into the mouse eye. And then we showed that we were able to modify a high amount of the DNA in the mouse. And this picture here just shows you that we were able to cut out that little piece of the DNA, a little piece of DNA in the PRPF31 gene. And then when we look at the function of, at the visual function in these animals, what we see is that they have reduced function. So we used ERG recordings to look at the function of the retina in the mice. ERG stands for electroretinograms. I bet a couple of you have had these in the clinic. And the way it works is they put an electrode on the surface of your eye, and then they flash a light in front of the eye. In response to that flash, your retina has an electrical impulse, and we can actually record that with the electrodes that are placed on the surface of the eye. So the gray lines there, which you see kind of a bump in the line, that is normal functioning after a flash of light in a mouse eye. And then the blue line is much flatter. It shows that in these animals where we edited out the PRPF31 gene, the retinal function is reduced. And that's true in both dark and light conditions. And then this is a picture of the mouse retina. So if you imagine taking an eye and slicing it like a loaf of bread, and then taking out like the middle slice of the bread and looking at it, that's kind of what we've done here. So this is a slice through the retina, and we can see all the different layers in the retina. And on the left side, that's a normal mouse. And then the right-hand picture, that's one of the mice that we edited to create this PRPF31 model. And what you see here is that the retina is a lot thinner, and you're missing a lot of blue dots. Each blue dot is a retinal cell. So it just goes to show that we've created this model that really closely recapitulates the symptoms of PRPF31. So now we have our mouse model. Obviously, what we want to do at this point is to show that a gene therapy can work in this animal model, which would give us evidence that the gene therapy is likely to work in patients in the clinic. And so what we did in these mice was actually to inject two viruses. So one virus carried in the genetic scissors that cuts out PRPF31, and then the other virus carries a healthy copy of PRPF31, which is shown in red here. And so this is that gene augmentation, that gene replacement that we talked about at the beginning. We're replacing the edited copy of PRPF31 with a healthy copy in order to treat the disease. And these pictures are, again, cross sections through the eye. So again, we've sliced like a loaf of bread, and we're looking at the sort of middle section here. And what you can see is that in the row on the top is the mouse that has the PRPF31 edited, and so the retina is a lot thinner, and you can see that this is a diseased retina. On the bottom is a section from an eye that was treated with the healthy copy of PRPF31. And what I hope you can appreciate in these pictures is that the retina on the bottom is much thicker. And so we've prevented those cells from dying off. And so this shows that the gene augmentation was successful in preventing retinal disease from developing. And then if we look at the function of the eyes that were injected with the therapy, again, here we're looking at these ERG traces, electroretinogram. So it shows a response in the eye to a flash of light. And so the blue lines show you the diseased eye, and the red lines show you the eye that was treated with PRPF31 gene therapy. And you can see that the amplitude of the trace, the height of the peak in the animals that was treated with PRPF31 is much higher. And then on the bottom, this is just shows you quantification over a bunch of different mice. We always want to see that it's going to work in more than just one animal. And so I hope what you see here, what you appreciate here, is that there's a better visual function in these animals that were treated with the PRPF31 gene therapy. So last thing I'm going to show you here before just summarizing and closing up is that, I think we've got really great evidence that we have a good mouse model. But one thing that I think would be helpful is to look at a large animal model before moving into the clinic. So I think we've all heard that, sometimes things we develop in mice in the lab don't translate well actually into people because mice are so different from people. And so one thing my group is working on is using the same type of approach where we're using these CRISPR molecular scissors to cut out PRPF31, but doing it actually in primates. And so we do this primate work because primates have eyes that are very similar to humans. And so if we're able to create a primate model of PRPF31 and show rescue in that, it's even stronger evidence that the therapies we're creating are actually going to work in people in the clinic. And so we did the same type of approach here. And what this is showing you is that we're able to deliver the CRISPR. This second, I shouldn't have clicked through so quickly, but this second chart here shows you between these blue lines that we were able to cut out the PRPF31 gene in monkey retina as well. And then these other pictures here show you that we're able to actually knock down or eliminate some of the PRPF31 in the monkey eye as well. So this is showing us evidence that we're moving towards creating a monkey model that we can also use to test these gene therapies even further and show even more evidence that it's going to work in patients. So just to wrap up here and summarize, showed you work from our lab showing that a CRISPR-based approach can be used to create mouse models of PRPF31 that model the main features of this disease. And on top of that, we did a gene therapy, we did a gene augmentation that rescued the retina. So it prevented the degeneration in the retina and helped to maintain function in these mice. And moving forward, we're working on creating primate models, but also always with our eye on the clinic and hoping to move this into the clinic and moving it towards patients as quickly as possible. And that brings me to the end of my presentation.
Thank you, guys. Thank you all. It's been very fun and very interesting for me to see the progress over the years that has happened, and particularly what I see coming out of Pittsburgh. But you've all been doing this a long time. Would you say that in these last few years we've seen more activity where there's every year more clinical trials coming on, and it seems to me that we've sort of reached a tipping point where we're starting to see an acceleration of these clinical trials coming to fruition. Is that safe to say?
Yeah, it's safe. At the same time, as I was trying to say, there are a lot of difficulties we still have to overcome. So it's not an easy, very straightforward path. There is a lot, and Leah was describing the steps, and this applies to a lot of these projects. You have to validate the safety, the efficiency, and it might be different from one gene to the other, but we are learning a lot. And if we remember the situation 10 years ago, 15 years ago, it's really dramatically improved in terms of efficiency, and the hopes are very high, but at the same time, we have to be careful.
Yeah, well, and your contributions certainly noted that there's a lot of work that's coming out of both Pittsburgh and Paris that contributes to some of that progress. I'll get to the questions from our viewers. We had a lot of people on today and a lot of questions. One question relates to AION. Is there any AION gene therapy research in progress?
Well, it's more like an ischemic condition, inflammatory ischemic condition. So not really currently, but it's more on the immunology and immunosuppression approach. So the only approach that could be relevant in the future is for patients who lost the optic nerve because of this condition, working on optic nerve regeneration, which is another topic we covered and we are happy to continue to cover. And it could use gene therapy to deliver the neurotrophic factors, but not exactly, it's not directly a genetic disease. And maybe there's a question about what type of condition. Currently, it's really focusing on genetic disease, although you could use gene therapy to deliver therapeutic proteins. And then this could apply to other conditions like vascular condition or age-related macular degeneration.
Right. So there's the actual therapy of using gene therapy techniques to deliver different ways to try to do regeneration in an area like the optic nerve, but there's also which is different than using a gene therapy to actually
Correct.
Adapt or correct a gene.
Yeah.
So how does an interested patient go about getting the genetic testing? Is there a process, and where can patients find more information to discuss with their care team?
Maybe Michelle?
Yeah, I can start with that one. So I think the most important part is really to have an evaluation with your clinician and find out whether or not genetic testing is recommended in your case. For instance, something like a retinal dystrophy, genetic testing would be, routinely recommended to patients, but there are other ocular indications or diagnoses that, as Dr. Sahel was just saying, don't necessarily have a genetic component that we know of. And so in those cases, maybe genetic testing would not be recommended. But for individuals with retinal dystrophy, for example, having an updated evaluation with your clinician to make sure that we really understand what we call the phenotype, or kind of the clinical presentation of your specific case before we move on to doing genetic testing, because that information really helps us to interpret the data that we get back from genetic testing. So I would say start with talking to your physician. Ultimately, they are who would order the genetic test. And then here at UPMC, it would be a genetic counselor that you meet with after that, who would walk you through the steps. As far as where you can go for more information, there's a lot of information on the Foundation Fighting Blindness website about genetic testing and a sponsored testing program that they have, where they help to cover the cost of testing for some patients. And we participate in that program. And then on the UPMC ophthalmology website as well, there's some information about the genetic counseling that is available through our department.
Terrific. And Dr. Sahel, you brought up the National Institutes of Health website as well, right? That's a source to find out whatever type of clinical trials are going on anywhere in the country.
Yeah, it's the clinicaltrials.gov website, but people should be careful. This site has also trials that are not scientific very valid. So it's a list of trials that are listed. Just a clue: anytime people are asked to pay for a clinical trial, we should not accept, because the clinical trial should be free. And this is something very important to know, that there are some so-called trials on this website that are actually commercial trials that are not really for the validated therapy, which is a bit annoying that these therapies are offered this way. But this trial lists a lot of information. We also have a lot of information on the website of the Bioengineering Foundation. And as Michelle was saying, the Foundation Fighting Blindness is providing a lot of information, and they are covering genetic testing for many patients through My Retina Tracker. Yeah.
Excellent.
To get involved, I'm seeing the question, to get involved in the trial, really, people should contact us, because we are leading many of these trials. And if we don't do it here, we know where to do it if it is relevant for the patient.
So are gene therapies typically covered by insurance? Do we know this yet?
So the trials are free again, so they are covered, and Luxturna is covered by many payers, but not all of them. And it's a process. So there is an authorization specialist. So we just went through that. Our goal is to make sure that everyone has access to therapy. And as you know, in the department, we have a strong focus on that. But most plans are covering it, but not all of them, unfortunately.
Yeah, it's a very new therapy, so this will have to play out over time.
It's very expensive. It's very expensive.
Yeah. So how many other clinics in the U.S. are currently offering gene therapy treatments? Do we have a handle on that?
So I think for Luxturna, it's 14.
17 is the final count for Luxturna, which again is the single FDA approved gene therapy. And then each of the individual clinical trials, which there are dozens of, are going to have specific sites across the US, probably less than that initially, because they try to kind of minimize the number of variables that are in place during a clinical trial. But as far as FDA approved gene therapies, Luxturna is the one example that we have right now, and there are 17 in the US that are spread kind of across the country.
And this includes Pittsburgh. We are a site for that. And we actually also have several ongoing clinical trials in Pittsburgh. Yeah.
So Leah and Michelle, what interested you in this type of work? And again, this is from one of our viewers. And how did you decide to make this your career?
I can take that first, I guess. I got interested in gene therapy when I was doing my PhD. So I got a PhD in neuroscience. And when you do a PhD, the school where I went, you typically rotate through 3 labs where you try some different things out. And I rotated in the lab of John Flannery, who also works in gene therapy. And I just fell in love with it. It ticks all the boxes for me in that it's very impactful. I like to work on something that's going to make a difference for people and contribute in some way. It's also very engineering. I like to make things, and I like to think about problems, and I've always liked working with my hands. And so it's a combination of a lot of different factors, but just ended up being a perfect fit for me.
Excellent. Okay, we're out of time here, but I'll read one. Well, I think we covered most even these other questions as well, as far as the conditions that can be treated and what we're starting to see in terms of what's happening in Pittsburgh. So listen, this is a very fast growing field. It's also something that we're very focused here on in Pittsburgh. I think it's safe to say that Pittsburgh is very invested as well with a lot of things that are happening in the city around biomanufacturing and other things that are a priority for Pittsburgh. So I think that gene therapy this is a good place for it to happen. And we expect that we'll be giving more updates on this in the future.
Yeah.
And I expect that there'll be that we'll hear again more positive news about the progress being made. I can also assure you that I know this team very well, and they are working extremely hard on trying to get the answers for all of you. So thank you all, and I think we will go ahead and end today's session, and stay tuned for the next edition of Sight and Sound Bites from the Eye and Ear Foundation.